All right, our class started. Welcome. And here we are. And we're going to have our exam on Thursday, our second exam. It'll be 52-point questions, so it's a very similar format to the first exam. Similar difficulty. Try to make it so you students don't run out of time while you're taking it. And it's going to cover chapter 18, 19, 20. 20s are really, all those chapters are really important chapters for us. It's the center of our course. So it's good we're having a final, or a second exam, and then 2 weeks after that, or less than that, we'll have our final. And the final covers everything. So we're pulling it all together. We will cover one more chapter after the final, and it will be the chapter on the nucleus, and that is chapter 11. And so we'll deal with that chapter next week, and then we'll have a final, and it'll cover some new material from the nucleus chapter, and then everything from our course, all the way back to the clay model videos that we started our course with. And then you have to write your term paper, and that's due to me by email a week from Monday, or a week from yesterday. And so that, we're focused on our exam right now, but then you can wrap up your term paper and get it to me. Remember that Riley Cott is the tutor for our course, and Riley's great. He's super nice, and he's super well informed. He took this course last year. He really understands how it all fits together. So you can contact Riley, and then he's going to visit our class one more time a week from Thursday and tell us one more shot about the CAR T-cell therapy. And it'll be more kind of the clinical aspect of it, of what is happening with the patient and what lies ahead for those kind of treatments, because it's just a really exciting new medicine. And so you'll learn molecular biology. So that's where we're at. And I want to show you this on the last day of classes at noon outside right there. The chemistry department is going to play its annual game against the biology department. And the biology department will win because we're the best, and that's what it's all about. And you should just come and play, and you don't have to have played kickball before or any other sport before. And you could play because it's really not competitive, even though we'll do a lot of trash talking because the chem department deserves that. And also, you can come watch, it'll just be fun on the last day of classes. So there's that. And so this is where we're at. I sent out an email last Thursday with a link from the recording saying, you know what, last Friday I was just going to make about an hour-long recording of what I was going to talk about today. And so I just sent that link out early, and that takes the pressure off and gives you a chance to have looked at that. I've essentially said everything I really want to say. We could talk about some new stuff today that will be important for the exam, but it won't be a ton of new stuff. It'll be stuff that gets brought up in, just discussing these topics. And so that's where we're at. And we have a lot of new stuff to talk about for chapter 20, and so that's why I did that, is that it's putting our course together. So we're in good shape. But you're learning all this stuff, so let's just see what kind of questions people have. And Catherine?
Could you just talk a little more about, so you have that list of characteristic properties of cancer cells, number 4, the secretion of proteases?
Yeah, so we learned about protease when we learned about caspase. And so there are lots of proteins that are enzymes, and proteins that are enzymes are going to catalyze some chemical reaction. And so we learned, for example, about kinases, right, like that, and we learned about phosphatases and stuff like that. And just with the caspases, we learned about another thing, and there are some enzymes that are called proteases. And their substrates are proteins, specific proteins, and their catalytic activity is to cleave that protein between some of its amino acids. And there are many different kinds of proteases. The ones that you most often would try to learn about initially would be the ones in our digestive system. So our pancreas releases bile, and it has proteases in it to help degrade the proteins in whatever food we eat, because we never use the proteins in the food we eat. Even if we're short on insulin in our body, we can't use pig insulin or cow insulin that we have to eat if we eat meat. Instead, our body, and really every animal or plant, every cell takes proteins it finds and it breaks them down into amino acids. And so there are some proteases that are involved in digestion, and they're really general proteases. They're just going to cut up the amino acids completely, just cut them all to individual amino acids. And so you could, it's not a formal list, but that's the way to start thinking about them is, oh yeah, there are a lot of proteases in digestion. They're secreted into our intestine, and they help really break down the proteins we eat. Then there's another set of proteases that are inside cells, and they're just degrading proteins all the time. And we didn't really talk about them directly, but we learned that in the Wnt signaling pathway for our first exam, there's a protein called beta-catenin. And beta-catenin is a transcription factor. And normally, in the absence of Wnt signaling, beta-catenin just gets degraded. So it's made and it just gets degraded. And that's because there are proteases inside the cell that are breaking down proteins, and they're just chewing them up. Those proteases are regulated like that. They need to be shown what protein to degrade. And so proteins are tagged for degradation. And we learned about beta-catenin. That would be one example of a protein that gets chewed up by proteases in the cell. And the role of Wnt as a signaling factor is to bind to frizzled, and then through a signal transduction pathway, those proteases are inhibited, beta-catenin is stabilized, can do its job, and beta-catenin is a co-activator that binds to transcription factor TCF. So some of this stuff, right, that what I just said was from chapter 17, and chapter 17 isn't on our exam, but it'll be on our final. So we want to start looping that stuff in and having that big picture, okay? So then, yep.
What did you write for A?
A is like digestive proteases. So those would be secreted into the extracellular environment and, breaking down the foods that are in our intestines, right? Really breaking them down all the way. And those are good ones to start to think about, because they are just going to break down proteins in a really sensible way. We're digesting them. We want just the amino acids. We want to get those amino acids into our system and use them for the proteins we need to synthesize, human proteins. And then B are proteases that are within a cell. Proteins are constantly being broken down. They get broken down at different rates, and sometimes the rates are regulated. So beta-catenin becomes a really interesting example of a protein that is unstable in the absence of Wnt signal and then stabilized. Another protein we talked about that gets degraded really fast is MYC. And we talked about that because MYC is a transcription factor that can activate cyclins and transcription and expression of cyclin proteins. And so it's going to be in the pathway to stimulate the cell cycle we learned about in 18. And MYC is really dangerous protein. And so the cell really regulates it carefully. The RNA, the MYC, from the MYC gene, the MYC mRNA, that's really unstable. It just gets degraded really fast. Then ribosomes don't want to synthesize MYC protein. They don't really like the MYC RNA. Then when MYC gets translated into protein, the MYC protein only lasts for like 10 or 15 minutes. It just gets chewed up inside the cell. So that's our second example, and it's regulation of degradation, and it's specific. So we're thinking about that. Okay? Then we talk about proteases specifically now because we want to talk about these caspases. And the caspases are proteases. Their substrates are proteins that they proteolytically cleave. So this process, just to use the word, is called proteolysis. So proteases stimulate chemical reactions that are involved in proteolysis. That's like protein lysis, and lysis means to break. And so caspases are the really effector proteins in apoptosis, and so we really want to understand those well. And there are orthologs of caspases in every eukaryotic cell. Definitely all plants and animals, and I think even, maybe there'd be some exception of some single-celled eukaryotes, but plants and animals do a lot of apoptosis. And caspases are the effector. They're the ones who really do the job. And so they have some targets. And we learned about 2 targets of caspases. One target that we learned about was this scramblase protein. And scramblase is the protein that can flip phospholipid to the outside of the cell, from the inner leaflet to the out leaflet. And so caspases are interesting proteases because they don't degrade the protein targets, they just clip them. So the scramblase is synthesized by as a pre-protein, a bigger pre-protein, and then it's always present in every one of our cells all the time, and it's inactive. And then when the caspases get activated, they proteolytically cleave scramblase, they activate it. The other one that we learned about is caspases also cleave DNases, deoxyribonucleases. Those are enzymes that break down, degrade DNA. So the caspases cleave the DNase as a pre-protein and activate it, and the cell's DNA just gets degraded like that. So those are 2 things we learned about, what is actually happening when a cell is dying. Like that, okay? And so that's the answer about proteases. Yep.
You said something about either the tumor or the protease being invasive. Is it more so
Oh yeah, sorry, we didn't get to that yet. So we learned about that. And then we want to answer this question number 4 about proteases. So that's great. So that's kind of our setup for that, okay? And so in these characteristics of cancer, I guess you, Catherine, said it was number 4 that I talked about, is that cancers release these proteases, and the proteases degrade proteins. And cancers, one thing that's talked about, we've mentioned it some in class, and you probably heard it just out, in your daily life. Cancers are progressive diseases. So that cancer gets more and more out of control. And you've probably heard about it in your daily life because cancers can be growing. It's a tumor, it's early, maybe we can get it, surgically remove the tumor and the patient will be fine. But then they'll say, oh, it's stage 4 or stage 5. It's an advanced stage. The cancer has spread. So that's an example of this idea that the cancer progresses. You could start out with a mutation and it's not even cancer yet. It's precancerous, like in a person that has a kind of a sustained infection of HPV. That virus is causing problems to the cell. It's not causing cancer, but it's moving the cell more toward growing out of control. So it could be precancerous, benign like that, and then there's a mutation. Maybe it's in regulation of MYC, so the cell starts to grow through the cell cycle too much. But it's still just in one place. Then it gets to be a bigger tumor, and so it needs to start doing things like it needs to start attracting blood vessels because it needs the oxygen and carbon dioxide gas exchange, and it needs to get nutrients, get rid of waste like that, So it's a progressive thing, and we've talked some about how cancers pick up other mutations. Sometimes they pick up mutations in DNA repair or DNA checkpoints, and that leads to just a more likelihood that there'll be more mutations that will get them to be progress toward being more problematic. So one of the things that can happen is cancer cells can progress where they start to secrete proteases. And these proteases are just outside the cell, and they just degrade proteins. And that's not normal. And what happens is the normal, healthy surrounding cells start to get their proteins chewed up, and that weakens their connection. one thing that's happening is that outside the cell, there's something called the extracellular matrix, and that's made of proteins and polysaccharides. Those proteins get chewed up. So that mesh that's sort of holding things together starts to get broken. And then these other neighboring healthy cells, they normally hold hands between each other with proteins. And these proteases from the cancer cells start to break those down. And so the tissue starts to be looser, and that provides more opportunity for the cancer to grow. And you probably have heard the idea that cancers can be invasive. They spread, and they can spread by having help with these proteases. So that's what's happening. That's the idea of these proteases, that it's part of this progression of cancer where now that cancer is starting to secrete these proteases, and it's starting to make the surrounding tissue sort of looser and more likely that the cancer could push out further into between these normal healthy cells and spread. Okay? So let's just look at this ABC list, okay, and say the proteases that we just talked about that are about cancer, which of the ABC categories do they fit into?
Probably A.
Huh?
Would it be like A, just choose of everything?
Yeah, because they're secreted, right? So A is like we think of these digestive proteases that are just secreted outside of the cell, and they're just going to chew up every protein they bump into. Definitely not C. These proteases have targeted substrates, and they're activating them. And definitely not B. Those proteases are inside the cell. So the proteases in number 4 in the cancer types are like in the category of like digestive proteases. They're not the same proteases, but they're like that. They're just digesting proteins outside of the cell, and that causes the tissue to be more able for the cell to push out in between, like that, and spread, invade. Yep. Yeah, so following up on that or other stuff.
Kind of in the same
Yeah.
area, would you be able to explain uncontrolled proliferation a little bit more?
Yep.
And like how it's different than
Sure.
like the precursor differentiation?
Sure. Yep. So that's a kind of a question that'd be more on the final, because we talked more about really the upstream regulation of cell proliferation, but there could be some of it on the second exam. And so our favorite would immediately go to talking about the normal healing of skin cells under a scab. And that involves the platelets secreting PDGF. PDGF binds to the PDGF receptor. It's a receptor tyrosine kinase. It autophosphorylates, and that causes it to bind to and activate RAS. RAS exchanges its GDP for GTP because RAS is a monomeric G protein, and when RAS is bound to GTP, it's active. RAS in turn activates RAF, MEK, ERK, ELK, like that, right? So it's on a pathway. Okay, out of control proliferation, our favorite example, but we could come up with several. Our favorite example is, well, when there's a mutation in RAS, it's in the coding region, the RAS protein that gets made is defective and can't hydrolyze the GTP as well as it should, so the RAS protein is just overactive. When it's overactive, it's less dependent on upstream growth factors like PDGF. So, under a scab, the cells are stimulated and they're proliferating in a controlled way. But when there's a mutation in a gene, like an oncogene like RAS, the cells are proliferating in an out of control way. Because they don't need the normal growth factors. Another way a cell could proliferate out of control would be if you have a mutation in a tumor suppressor, like retinoblastoma, P53. They're supposed to be putting the brakes on, so that's controlled proliferation. If there's a mutation in the tumor suppressor, then it's out of control proliferation. Then there would be more examples of oncogenes that are proliferating in an out of control way that would be comparable to our story of RAS. Oncogenes fit in a normal, healthy person, they fit in normal controlled proliferation examples. And when there are mutations in oncogenes, then the regulation is lost and the cell's proliferating out of control. Another example would be BCL2, where the cell will be proliferating out of control and failing to go through apoptosis. So not just going out of control, but failing to go through out of control is really kind of the same story as growing, proliferating out of control. Yep.
So when we were talking about inhibiting the inhibitor and apoptosis, is this just about BCL2 inhibiting pro-apoptotic proteins?
No. BCL2 is an anti-apoptotic protein that is inhibiting caspases. So the first thing to say is learn about caspases. And caspases do the biochemistry of cell death. They clip these proteins that are just going to kill the cell, right? And so the first thing to say is that they're the killing proteins. The second thing to say is they're in every one of our cells right now. The third thing to say is they're inhibited by proteins like BCL2. BCL2 is an inhibitor of caspases. So that's the situation in every one of our cells right now. Our cells are not dying from apoptosis because they're inhibited by BCL2 and other proteins, but they're inhibited. They're ready to die, like that, okay? So apoptosis is a story of blocking BCL2. And one way you'd say that is we're going to inhibit the inhibitor. And that's only a little bit complicated, and I don't think it's complicated when we say it in that order, right? Our cells are all poised to die right now. And they're not dying because the caspases are present but inhibited. And so programmed cell death apoptosis is overcoming that inhibition or inhibiting the inhibitor. So some of that occurs because other BCL2 gene family members inhibit BCL2. And as a result, those proteins are pro-apoptotic because they're inhibiting the inhibition of apoptosis. Yep. Yep.
So when you say that a mutation in BCL2 can lead to cancer, like why does that not just lead to like a lot of apoptosis? Because it can no longer.
Yeah, because that example that we gave, which is in non-Hodgkin's lymphoma, is that there is a mutation in BCL2. And it's just really good for us to fully step up to this level course and say, well, you remember in intro bio, you learned about mutations. Like, there could be a person who has sickle cell anemia, and that's the most common intro biology example of mutation. The person is making a defective form of hemoglobin. And so that's how you initially learned about mutations, is they're making an altered form of the protein. And what we say here is, those mutations that make an altered protein must be in the coding region of a coding gene to make an altered protein, because the transcription unit is the part of the DNA where mutations could occur, and it makes the codes for the product. And if we have an altered product, like a mutation in RAS protein or hemoglobin sickle cell, it must be a mutation in the transcription unit coding region. And so that's the one, like, if you are learning biology, that's when you think about a mutation, you really typically think of that, right? BCL2 mutation, oh, it messed up, just I 100% agree, oh, it's messed up. But we learned that genes are made of two parts, and those parts are both DNA, so mutations can occur in DNA. And so there are other types of mutations that can affect a gene. The mutations can either be in the transcription unit and affect the product, or they can be in the 6 regulatory elements and affect the where, when, and how much is made. And so that's the mutation that BCL2 has. And our example is non-Hodgkin's lymphoma, where there's a chromosomal translocation. And that's a type of mutation that's a dramatic alteration of a gene. And in the examples we talked about in our course, the chromosomal translocations involve taking the regulatory region, the cis regulatory elements of one gene, and accidentally connecting that DNA to the transcription unit of another gene. And so in non-Hodgkin's lymphoma, the mutation is that we have a chromosomal translocation between an antibody gene that is normally expressed at very high levels in B cells, which are white blood cells that are circulating in our blood, and that regulatory region for the antibody gene accidentally got joined to the transcription unit of BCL2. And so we should be able to draw that all day long. We drew it on the first exam, we have to draw it on the second exam, because it'll be like, describe a translocation, draw a translocation, and you either draw BCL2 translocated with the antibody gene, or we also learned about some other cancer, Burkitt's lymphoma maybe, that's got another antibody gene joined to MYC. In both those cases, it's a mutation. It's a mutation in BCL2 or a mutation in MYC. But the transcription unit is normal, it's wild type, and it makes normal BCL2 or MYC, it's just too much by a lot. And so that's how BCL2 can lead to cancer, is too much inhibitor. As a result, you can't inhibit the inhibitor because there's too much of it. Our book shows how BCL2 family members that are pro-apoptotic, like PUMA and BAD BIM, several of those work by binding directly to BCL2. And so just think about that for a minute, right? Here's this amount of BCL2 in the cell, and to overcome that inhibition, we're going to directly bind to it. What you are saying is that there's actual molarity, a concentration of BCL2 in the cell. And we need to make a higher molarity of our pro-apoptotic protein, PUMA or whatever it is, in order to bind all the BCL2 and inhibit it. And the chromosomal translocation is increasing the molarity of BCL2 so that you just don't make enough PUMA to inhibit it. You inhibit some of it, but you've made too much. So you can inhibit, 1,000 molecules of BCL2, but you've got 75,000 in the cell. So that's what's happening with BCL2. Yep. And it doesn't cause cancer. It just is that if the cells start to grow out of control, they fail to go through apoptosis. Yep.
Is the opposite true for these examples where, say, the antibody regulatory region is switched with the MYC regulatory region, so that in the case of Burkitt's lymphoma, you're making MYC, but would you be, for the genes that are coding for MYC, be making the antibody?
Yeah, so that's true, that in a chromosomal translocation, there are 2 genes involved, and we actually create 2 mutated genes. In the case of the BCL2, we are creating a mutated form of BCL2, and we're making a second gene that's a mutated form of the antibody gene. And that gene has issues, but not ones that we focus on. In that example, it would have the BCL2 regulatory region upstream of the antibody protein. And so you'd be making some antibody protein regulated by BCL2, so you'd make some, but it would not be problematic because the cell is supposed to be making antibody like that. The other thing about that is that in these cells, they're always diploid, and so even though we have a mutated form of the antibody gene, the person, that could have been their gene from their mom, and they've got a version from their dad too. And so that's always a factor in this, in that probably, you know, they're probably, in that sense, making a fairly healthy amount of antibody, because it's like half as much as normal. Yep.
Can we go over the MCM and the P27 again?
Yep. Yeah. So the first thing that we learned about MCM is this, and is that here's the DNA, and MCM protein binds at locations where DNA replication should start. I should say begin, so we don't confuse it with that concept of, start. So DNA replication begins here. The chromosomes are so long in our body that we can't just have DNA polymerase start at one end of the chromosome and go to the other end, because it would take way too long. Instead, many DNA polymerases, many of them, have to bind all along the chromosomes and help. And the official term for these sites they're called the origin of replication. And of course, they're always there, and the enzyme DNA polymerase is always in the cell, because there could be some DNA damage and it needs to help fix it. And so this process of finding the origin of replication, and which occurs during S phase, that's really highly regulated, because in G1 or other times, we don't want the DNA polymerase to bind to the origin of replication and start copying it, because we're not in S phase. We don't want that happening. So there are lots of stuff that happens to regulate that. And one of them are these MCM proteins. And the function of MCM protein is to bind near origins of replication and show DNA polymerase where to start. Just as a little side note, sometimes this helps students, sometimes it confuses them. Sometimes it's nice to know the name of a protein. This is called mini chromosome maintenance. That's kind of a memorable name. And what scientists did is they just made an artificial chromosome years ago. So they just made a small mini artificial chromosome with one origin of replication, and they asked, what proteins do we need to keep this little artificial chromosome maintained? It's got to get replicated or it'll get lost. And so they just made that, and then they started studying that, like in one lab or two labs. That was their thing. They're like, we made this little mini chromosome, and we just see what it takes to keep it, right? They would have put a gene on it, like GFP, something to make it glow, and say, yeah, it's being maintained. The cells are still glowing. And then they'd start looking for mutations where, they're no longer maintained, right? And they found a mutation in a gene for MCM, like that. So they call it mini chromosome maintenance. That's why they call it that. So there are proteins that have cute names like that. It really derived from the history of how they ever figured out what that does, right? And our molecular biologists love to talk about that. My first year in graduate school, my advisor taught a course, and he just talked about the experiments like that. He didn't talk about the summary figures. He just talked about mini chromosome maintenance like that. So that's our first one, MCM. And it's involved in S phase, like that. And so we learned two things about it. We're just going to complete the story of MCM. And one is that to get its job done, MCM has to get phosphorylated. And so MCM is a target of the start CDKs. And I think Tiva's asking about, we have a little list of 3 things that we learned that CDKs phosphorylate, and that's a really good one. Because some of them are already present, but they're not active, they're not doing their job. And the purpose, when we say, oh, the light is turning green, what we're literally saying is biochemically, we are moving into S phase. So we need to activate these to show, so they do their job. The second thing we learned about MCM is that, of course, MCM, since they're proteins, there's an MCM gene. And I'm just going to erase this and draw it again. So the second thing we learned about MCM is that, here's the DNA, and here's the MCM gene, like that. You can make it, and if we, in S phase, we need a lot of this. We have a little bit in a cell, and we're going to phosphorylate that. That's going to get things going, but we also need way more. There are many origins of replication. I don't know, on a chromosome, how many are there? A couple thousand per chromosome? There are a lot. So we just need a lot of MCM. And that's a thing that we talked about, and a student used the word, we got to stockpile these. When we're going to roll into S phase, when we turn the light green, we got to phosphorylate things to activate them, but we probably also need more of them. So we should be able to draw on the exam a drawing of the MCM gene in G1. And in G1, the MCM gene looks like this, where bound to a cis regulatory element, an enhancer, is this protein E2F. And E2F is a transcription factor. And anytime there's a transcription factor in a cell, there's lots of molecules of it. There's lots of molecules of E2F during G1, and they're bound to several target genes, or E2F response genes, right? And this is one of them. One of them is MCM. During G1, we don't want these genes made. The other genes include targets that make enzymes that synthesize the AGCT nucleotides. We want that during S phase, but we don't need much of that during G1. A little bit for DNA repair, but we need a lot, like that. Here's another gene, DNA polymerase itself. We just need a lot of DNA polymerase, folks. Let's go. So that's that, again, that stockpiling notion of we just need to make a bunch of these, come on, right? Okay? But in G1, no. We mostly need this gene off. Maybe we have a little bit of this made, but mostly we want it off. So in G1, we would really like to have a repressor, which will make red. And what is that protein? It's RB, or retinoblastoma. And retinoblastoma is bound to E2F and repressing it in G1. RB, or retinoblastoma, is a co-repressor. A co-repressor is a transcription factor that does not bind to the DNA directly. It doesn't have a DNA binding domain. It binds to some other transcription factor, like E2F, that does have a DNA binding domain, is bound to enhancer, bound to cis regulatory elements. Okay? So that's the setup. There's RB, and you're like, okay, cool, right on, So to go into S phase, one of the things is, remember on our list, well, we want to phosphorylate that MCM. So there's already a little bit of MCM, and the CDKs are phosphorylating them. We also want a lot more MCM made. And so another target of CDKs are in S phase. CDKs add phosphates to MCM, they also add phosphates to retinoblastoma, and they inactivate retinoblastoma. Okay? And when it's inactivated, what do? We removed an inhibitor. So we have that same situation where essentially we're inhibiting an inhibitor, and you know, that sounds like a double negative, but there it is, right? It's, yeah, I get what's going on there. RB in G1 is inhibiting, and RB in S phase is not, because it got phosphorylated by CDKs. So that's a second example of what CDKs target, right? We saw them phosphorylating MCM. Here they're phosphorylating RB, which leads to more MCM protein being made, cool, but also lots of other targets. That brings up this HPV, human papillomavirus, is that what it stands for? Right, that thing that causes genital warts. We learned about that, and in chapter 1 we see one of the gene products, one of the proteins made by that virus, inhibits RB, and it releases the E2F, and we just start stockpiling it. Who wants that? The virus is a DNA virus, and it's like, oh sweet. Just look at that, like that's just an Achilles heel for that DNA virus and infection. If the virus could evolve through natural selection, its own gene that makes a protein to inhibit RB, go like that. Jerry might pointed out that inhibiting it, you don't really go into S phase. It's not really that the light is fully green, but you just do this stockpiling thing, right? You just start stockpiling this stuff that is exactly what the virus needs.
Stockpiles of E2F.
It removes RB, so E2F can do its job, and E2F is a transcription factor that activates several target genes. And those target genes make products, and the products are things like MCM, enzymes that synthesize the AGCT nucleotides, DNA polymerase, and others. Those are the things that get stockpiled.
So E2F is free to do its job.
Yep, and its job is a transcription factor to activate target genes, and those target genes products are things that we need for S phase. And so that's the stockpiling. The idea of we need a lot of MCM, folks. Let's go, like that. Yep, and that's that stockpiling concept. And so that's another one, okay, like that. And then these CDKs at start, they're phosphorylating a lot of things. And we learned about MCM, because that's a good one for us to see. Protein's already present at a certain level, low level, and it needs to be phosphorylated to get its job done. So it does that. The CDKs, it's also see this, they phosphorylate and inhibit this really famous retinoblastoma tumor suppressor. And that allows lots of other proteins to be made, including even more MCM. And what happens as more MCM gets made is the CDKs are just phosphorylating, right? It's just this factory. We're opening a DNA synthesis factory like that. Yep, right? Yeah, okay, so now, remember that little girl that I knew whose eye was closed, I think because she had it surgically removed because of what her dad said to me off to the side. She's a carrier of the RB gene, which means she got a gene from her mom and dad, and I think it means she inherited a mutated version, like that, okay? And as a result, wasn't getting this job done very well, and then I think what happened in her body is she accidentally picked up a mutation in the other copy of the RB gene, right? So I think she was born like a carrier of this, where she got a normal version from, say, mom, and a broken version from dad. And if she got a mutation in mom's, then she doesn't have this. And it's like this thing, right? It's like where she doesn't go into S phase, she just starts stockpiling things. And so it's a way that start is complicated. If she starts stockpiling things, that just sets it up where, oh boy, you know, if you get another mutation that turns that light green, you're gonna be rocking and rolling, you know. There are lots of checks and balances in start, and if you start removing some of them, you're in trouble, okay? So, I won't ask these questions on exam, but where's the mutation in that RB gene?
In the coding region.
So, good chance it's in the coding region, right? Because when you make a mutation, it's random, so it's a loss of function, right? So she's just making a defective version of RB. It's just not getting the job done. It could be in the regulatory region also, where she's just not making RB, right? So in that case, right, it could be either one, but it's definitely a mutation in RB, right? Like that. And we don't know her situation. We don't know her family situation, but a genetic counselor would do some tests and counsel the family. Talk to them. She had an older sister, right? She had parents. Talk to them about, who are your family have this issue and what is the detail of the issue? Maybe she had mutations in both RBs and she just needed a mutation in the MYC gene or something like that. So she was just, a ticking time bomb like that. And why in her eye? Well, we talked about that in our course, right? The woman with skin cancer has a mutation in that PDGF gene, and it's skin cancer because PDGF plays a normal role in regulating skin growth. So when we talk about skin cancer, the person most likely that has skin cancer has mutation in genes that are involved in the normal growth regulation of skin. We saw that KIT gene, that's involved in gastrointestinal stromal tumors. It must be that KIT is normally involved in the growth of those cells, not so much in the skin. So KIT is mutated, and we find it at high frequency of that specific type of gastrointestinal stromal tumor. Not every kind of intestinal tumor, but that type, like that, right? We saw that with breast cancer and the role of the estrogen receptor. Well, the estrogen receptor is present in breast cells because in adolescents, those cells are sensitive to estrogen. So it's part of their normal growth control. And, if you have a mutation in the estrogen receptor, the cells that would be affected, cancerous, would be ones that are normally regulated like that. So that's kind of a typical idea that we think about like that. And it turns out apparently that in retinoblastoma, that must play an especially sort of sensitive role in the eye. Maybe there are just naturally not as many checks and balances or something in the eye, because that's why they call it retinoblastomas. That's where they first discovered it, in a tumor that's called retinoblastoma, a tumor that grows in the eye. Yeah, it's in all our cells, right? That's really common in science. You find it first here and you're like, oh my gosh, plays a role in general. Just found it here first. Yeah? Yeah, so there's just a lot of memorable things to piece this together. And going back, I think, to Tiva's question about the MCM, we found MCM story where the MCM protein needs to be phosphorylated. And then the second story is we make more MCM. So those are two really good stories about the role of CDKs. So in the role of CDKs, the targets we learned about phosphorylation of MCMs and activation of them. We learned about phosphorylation of retinoblastoma and inactivation of it, right? Because targets can be either activated, MCM, or inactivated by phosphorylation. And you just have to deal with them on a case-by-case basis. And the third one, just to finish, that was P27. And we learned that P27 gets phosphorylated by CDKs, and that's a good one because it brings up this principle of positive feedback. P27 is a small inhibitory protein, and if that's present and some CDKs get activated, the CDKs phosphorylate P27 to help further activate more of the CDKs. And that's an example of checks and balances, the idea of positive feedback, negative feedback. Those are checks and balances concepts. So, we're just carefully picking and choosing, right? CDKs have a bunch of targets, and we don't want to, overwhelm you with just a long list, right, like that. So let's pick and choose, and those are three good ones, like that, Yep. And
With the RB, so you're saying like the retinoblastoma is mutated in some way, and with this, like the example of the little girl,
Yep.
How is
So
that
in her
not
case,
there?
what would happen is she had a functional eye, fine eye. Then she started growing a tumor in it, right? So at some point, the growth of a tumor just inhibits the normal function of the tissue that it's growing in, like that. You know, when somebody has stomach cancer or brain cancer, the growth of it just starts to disturb the normal function of it. So she just had a growth in her eye, like that, a chunk in her eye. Yeah. And I was and we in class I just said, look, let's learn a list of the CDK targets at start. And if you think about it, the way that like a teacher is teaching something, those strategies are good ways to learn something. And so we saw that at a couple of times here in the material for the second exam, right? Where we talked about some stuff, and then we said, at some point I said, no, let's just make the list, right? So let's just do that. So these are the CDKs at start, and just the 3 targets we learned about, and you have to understand there are more. Start is complicated, right? Like that. Okay, so let's just go over to the G2 to M transition, the DNA damage checkpoint, just prior to the start of M phase. It's a different CDK there, CDK1, we don't need to know the numbers, but it's a different CDK. And it's an effector. It phosphorylates targets, and the cell is in M phase. So what are the 2 targets that we learned about that CDK and the G2 to M? What are 2 targets that we learned that it phosphorylates?
Is it B53 and
No. See, that's good, because we have these 2 different phases that we're talking about, and P53 is at start. So we're at a different place in the cell cycle.
The nuclear membrane?
The nuclear membrane, so it,
And the chromatin.
yeah, and we went over there because we said, this S phase, the S start, there's a lot of stuff going on there, and we're very invested in our course, right? Because we're looking at growth regulation, normal growth regulation, and so we talked about it a lot. There's start, the G1 to S transition. And then we went over here and we just talked about it a little bit, but it's just a really important a package. So at the G2 to M, we activate this CDK, and that means it's going to phosphorylate targets. And it's just satisfying to say, well, what are its targets? And the answer would be, its targets would be proteins that play characteristic functions during M phase. CDK is active throughout M phase, so what are some things that happen at M phase? And one is, in lots of cells, in human cells, for example, the nucleus breaks down. And you can see that under a microscope. And it's nice, and for a cell biologist, satisfying. You're like, we gotta get the nucleus broken down, because we got to get these chromosomes organized and get them lined up and pull them apart. The nucleus is kind of in our way, so let's just break it down, right? And so that's just a satisfying concept. And on our exam, we're going to talk a little bit about the different phases of mitosis and what happens in them, right? So this is a good one, right? And so here's the nucleus. That's the nuclear membrane. And the nucleus is always kind of a spherical shape. And the reason it's a spherical shape is because on the inner surface of the nuclear membrane is a mesh of proteins called nuclear lamins. And raise your hand if you know what a geodesic dome is. Raise your hand if you know what a buckyball is. Those are structures like a soccer ball. A geodesic dome could be like a building, like a house. A buckyball is a little carbon nanoparticle. And both of them look a lot like a soccer ball. where they're just assembled by little triangles and they make spheres. And that's what nuclear lamins are. They just are proteins that assemble into a spherical shape. And CDK phosphorylates them, and during M phase, the nuclear lamins disassemble. And they're just scattered around in the cytoplasm of the cell because they're all phosphorylated. And the nuclear membrane breaks down to the little droplets of membrane because it doesn't have that structure to hold it up. Nuclear lamins are a little bit like like tent stakes, where you got your tent, the nuclear membrane, the tent canvas is just laying on the ground, and you put in all the stakes, and you go, look at our tent, like that. Okay, so that's one of the targets we learned about, and it's just such a satisfying target, for a person to learn about, because you're like, yeah. At the end of M phase, the CDK gets inactivated and the nucleus reforms. The two nuclei form. Cool. And that's where these people, who are biochemists, molecular biologists are part biochemist, part geneticist, and the biochemistry part of the molecular biologists are just like giving each other high five, just being like, look, right? That there's no magic in this. We get down to that molecular level, it just all fits together. Okay, so then Brooklyn said another target we learned about, and the other target we learned about was the histones, which we learned about in the first exam. They're parts of nucleosomes, and the histones get phosphorylated, and that causes these nucleosomes to pack tighter. The chromosomes condense and become visible, which is another characteristic feature of M phase, is in prophase, the first phase of mitosis, the chromosomes condense and become visible. And
I
they're
had condensins and cohesions for that. Is that the same thing?
no, so those we talked a little bit about. Yes. We didn't talk about them. They're also involved in that. I just was a little bit confused, but yeah, they're part of it. I won't mention why I hesitated, but yes,
Okay.
yes, part of the story. Yeah. So, and I should say that, right? For the CDK1, we just learned of 2 targets, just 2. There are more phosphorylation targets. A bunch of stuff happens, and the CDK does that. So that's really important, that you don't need to know it, but just in principle, that there are other targets. Yeah, Eva?
CDK phosphorylates the histones.
Yep. In lab class, we did immunocytochemistry. We used an antibody that would stick to phosphorylated histone. It would only identify that. The histones are present all the time because they're helping package the chromosomes into nucleosomes all the time. But they're only phosphorylated in M phase, and they're phosphorylated throughout M phase because CDK1 is active all throughout M phase. At the end of M phase, telophase, the chromosomes decondense. Why are they decondensing? The answer is because the CDK got inactivated. Every kinase has a complementary phosphatase that removes the phosphates. There must be a phosphatase in there removing them and allowing the chromosomes to decondense at the end of M phase, and they disappear. They're still there, but they just spread out so much in the newly forming nuclei that you don't see them. Let's see, Ellie and then Brooklyn.
What is the purpose of them getting phosphorylated this time?
Condense. And I think you would say it's kind of a accounting or bookkeeping function where we're like, we need to get these chromosomes organized, folks. They're all spread out, kind of tangled and stuff, and we need to get them all condensed and then separate them, right? Like that. Like you're packing for a trip, you want to take your favorite clothes, but they're scattered all over your room. So you're like, we better fold up these clothes. Sometimes I wish I could talk like the comedian Nate Bargatze. Gotta fold up these clothes. Can't do it. Gotta fold up these clothes. Get them packed. We gotta pack them up. Get it going. Our trip. So first step, fold them, get them organized, then put them in. Just, like that. Sorry, he's pretty good. He has a great tone. So that, yes. Okay, then Brooklyn.
So yeah, the book goes into pretty great detail about the condensations and cohesions.
Cohesions, yeah. Yeah.
rather than the histones. Should
Yeah.
we focus more on the histone story for the
For
exam?
sure, anything, it's talked about in class, especially if we talk about it multiple times, then it's just like slam dunk. Like when I write questions on the exam, I'm like, these are easy questions, because we talked about them in class several times, right? Those are the easiest questions on the exam. So those histones on the exam, I would be like, okay, this is one that they're just gonna be like, yay, I'm going great, right? Like that. So cohesions and cadensons, right, the book talks about them a bunch, and maybe there would be a question, on the exam, and I would say that's the hard question, right? They're just gonna be a small number of exam questions where we didn't talk about them in class at all, yeah? But they would be ones that the book talks about, right? And you're like, yeah, I can see where that would be, that was a whole page on that, like that, yep, yep.
So we talked about the cell cycle and the DNA damage checkpoint.
Yep.
And we know that the checkpoint one inhibits the activation of CDK1.
Yep.
How is CDC25K role in this?
Yep, so that's, so we were just talking about this CDK1, and when it gets activated, the cell goes into M phase, just by definition. What you see under a microscope happening during M phase, it's CDK that's doing the biochemistry of that, activating those. And so we're gonna go into M phase if CDK1 is active. If there's DNA damage, don't go in M phase. Wait and repair it. And that's really satisfying. If we have damaged DNA, we should not be dividing the DNA. We should fix it, get it fixed, and then divide the DNA. So it's just really logically satisfying thing. Wait, okay? So the way the cell waits is there are two inhibitory phosphates that get added to the CDK, and they need to get removed. And they get removed by CDC25 phosphatase. When there's DNA damage, CHEK1 kinase gets activated, and phosphorylates CDC25 and inactivates that. And we don't activate it like that. There are, in our bodies, in a human, there are 600 different kinases, right? And so, there's lots of these stories of like, oh, DNA damage causes CHEK1 to be activated, CHEK1 kinase phosphorylates CDC25 and inactivates it, preventing it from removing inhibitory phosphates from the CDK. So you gotta, write that out, draw it out, to say it, to get that. This is another example of that inhibitor, activator, inhibitor, and it's just like, okay, it sounds like a double negative, but it's just, how it gets regulated. Yes, Hannah?
Is there a third one? Are those targets of the CDK, or is it just two?
Those are the two we emphasized, right? Because they're so satisfying. You're like, oh, okay, yeah.
We also talk about polo like an aurora.
Yes, you're right. So we did briefly. So when CDK gets activated, our book, and in class we wanted to emphasize, there are a lot of things that need to get done, right? Under a microscope, a lot is happening when a cell goes into M phase. So there must be a lot of things that need to be phosphorylated. And so one of the things CDK does, CDK1 is, at the G2 to M, is it phosphorylates helper kinases called polo and aurora. And why would we learn that? That sounds kind of goofy. Oh, you need some help. Just to underscore this principle of, we got a lot of work to do. When CDK1 gets activated, there's a lot of targets. There are a lot of histones just in this, we got to phosphorylate just a lot of histones. There are a lot to phosphorylate, lots of nuclear lamins to phosphorylate. But then there are lots of other targets. And sometimes the targets, there are many, many molecules of them. We got to phosphorylate them all, like that. So yeah, so that's why, for the book, right, a lot of times if you ask the author of a book, why did you put that in the book? The author says, well, because it's true, like that. That's their thing. In class, it's like, class has to be so stripped down and focused, it's because it's revealing a principle, right? And so why do we talk about aurora and polo in class? Because it just helps you see, there must be a lot of phosphorylation that needs to get done. CDK needs helpers to help get it done, like that. Yep. So true, yes, we learned about those as targets also. Yep. And we're doing great. Of course, we're not gonna have a concept chat today because we're chatting. Yep. But we have some time. We have another 10 minutes to go, or 5 minutes, I guess, to go. Let me just insert before you lose track of that. And I have, on Wednesday, I don't have, tomorrow I don't really have very much scheduled. So we could meet in person, or we could meet by Zoom. I can meet with multiple students, right? You might have a study partner and you'd be like, we should meet together, like that. That's also, I think I mentioned, maybe not, that when I was in college, there's no chance I ever went to talk to a professor. There is zero chance of that. And one reason is because unfortunately I had these friends who were jerks who yelled my nickname out, and so no professor wanted to talk to me either. Yeah, and yeah, I was not in favored status when I was in college. That's fine. I went barefoot my senior year in college in Minnesota. Yeah. Just because I'm like, Easter's coming, and they're like, Dave, it's October. So anyway, so I wouldn't talk to them. So I want to encourage you, we could Zoom, that's a pretty safe situation. Or you're going to have a friend or friends come, like that, right? We can talk, right? And yeah, I'm not, it's not scary. So let me mention that. Then let's see, was there a question or more questions people have? So we can plan times to talk. You can send me emails. I actually get several emails, and I try to answer those. If I got a bunch of questions, I can make another little answer for everybody, a little mini video, right? Where there's, oh gosh, this is going to take some time to explain that. Make a little mini video. So keep your eyes open for that tomorrow too. I might send a link and be like, look, I'm just going to add this, because I realize we failed to explain this properly, and it's confusing. So probably like that. So that could happen too. So check your email, but it probably won't, but it could happen. Just be aware of that. And yeah, but we can do a couple more questions right now. Awesome, I'll hand out the exam. Here it is. Oh, we can't hand the exam out, right? Yeah, because that's a great way to just drag your heels. I would do that on test day. I'd just be like, just before you hand it out, can I just say, you're going to want to have us put our name on the exam?
Is that worth a point or two?
Yeah. Yeah.
Do you mind explaining, so like one of the 8 things that you listed for like cancer cell
Yes.
functions was, I guess like 6, 7, and 8, I feel like there wasn't like a lot of explanation there, and I was just wondering if we should know, well I guess 6 and 8, so abnormal differentiation.
Yeah, let's see if I can find that. I had a thing where I had that right here. Let me just scroll and see if I can find that list. Well, yeah, there it is. And somehow it saved my writing. Oh, yeah, 6, 7, 8. So 7 is chapter 19.
Yep, BCL2.
Right?
I guess. Like,
BCL2 would be a good example of that, but there would be lots of that where it's not just uncontrolled proliferation, but it's also a failure to go through apoptosis. Because we emphasize, when cells are stimulated to grow, they're always also stimulated to consider apoptosis. Sometimes growth factors stimulate pro-apoptotic factors, sometimes anti-apoptotic factors, so it's complicated. But that's a hallmark feature of cancer. Chapter 19, the apoptosis part of that. So that's that. That's a package. Then 6, abnormal differentiation. There was just this one example of it that was provided, but it was a satisfying example. Okay? And it's in certain types of leukemia. And what happens is that every day of our lives, we have stem cells in our bone marrow that are going through the cell cycle, proliferating, dividing asymmetrically. So we have self-renewing stem cells being maintained in our bone marrow for our whole life, but just continuously churning out these precursor cells. And the precursor cells develop into all the different blood cells we have. We have red blood cells, we have white blood cells, the white blood cells that come in a variety of different types, B cells and T cells, a variety of different T cells. So there's quite a bit of development that's occurring in our bone marrow right now. Those cells continue to grow and divide, but then they reach that point where they're going to have their function and they differentiate. And when cells differentiate, they really typically leave the cell cycle forever. They sit at the red light and are in G0. So that's what's supposed to happen. And so there's an example of leukemia where the problem is different, and it's just a little example, but it should be satisfying to us. We could have oncogenes, we could have tumor suppressors, and programmed cell death factors. We've learned about those. And this is just a little different kind of story about cancer. In this case, what's happened is not out of control growth, not a really failure to go through apoptosis like that, but what happened is the cells just don't differentiate. So they stay in that precursor form, and they just keep dividing like that. And that's cancer. You know, we have out of control proliferation through this other mechanism. And, you know, we've learned the principles, and then this is kind of like an exception to the rule, where you're like, are there mutations in oncogenes? No. You know, apoptotic vectors? You know, no, like that, right? It's just that they fail to differentiate, and as a result, they stay in the precursor state, and they just keep proliferating, just proliferating more and more. Does that make sense?
So you're saying like there isn't a mutation that's causing that, it's just a
There is a mutation that's blocking their differentiation. So they have a mutation, and the mutation is they just fail to go through that normal differentiation, like that. And so they stay in that precursor state. The precursor state is in the cell cycle. You know, they have the right turning green, and they're just like, this is going to keep growing. Seems to be, you know, like that. Where it's like the person in college who has 150,000 credits. And they're like, are you going to differentiate? You're going to graduate and get a job? And they're like, oh, no, I'm just taking more classes, right? Like that, forever, right? And they're out of control. Yep, like that. Yes, so that's 6. It's a little package, and it's kind of like, oh, here's an exception to the rules we've learned. And that's not problematic. It's actually satisfying because it helps us appreciate normal development and how it can be disrupted. And then this capacity for unlimited replication, that's just one that's the most satisfying in the video, I say. The most satisfying example of that is if you ever take a cancer cell and you just take it out of the body, like you know, do surgery to remove the tumor and you keep it sterile, and you put the cells in a dish and you give them nutrients and keep them sterile, they'll just grow forever. They're immortal. There's a really famous example of that. There are many, many examples of that. There's a famous example of a woman named Henrietta Leakes who lived in the United States back in the 1950s, and they took out a tumor from her, and they didn't even tell her. They're just like, you got that tumor out, you're good to go. And they just kept it in a dish and grew them, and they call them HeLa cells, and they just keep them growing. So that's the best way to think about unlimited replication. Other cells, healthy, normal cells, they have limited replication. We're normal, our cells are immortal. Each one of our cells are mortal, but cancer's not. They just keep dividing. That's one part of it, is they just keep growing, they're immortal. The other part is actually one of the earlier ones, and that is that they have this reduced cell adhesion molecules. So not only are they immortal, but in a dish, they just keep spreading. And that's this thing where you're like, I don't like that. It looks bad, right? And you're like, yeah, it's a cancer cell, right? Like that. Normal cells in a dish, not only are they immortal, but just in that dish, they'll fill up the dish and they'll just stop growing because they're dependent on these cell adhesion molecules in this process called contact inhibition. We have to stop here.
I do have a question.
Yes. And so you could hang out and ask some questions, you know, and if we necessarily, we'll drift into the hall. But people can pack up and head out, and I'll send